Hi, everyone. Thanks for joining today. We'll get started on the hour. Hello, everyone, and thank you for joining today. Welcome to the NOAA Omics Seminar Series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and posted on our Omics website. There should be a link to the website in the chat now. As participants, you are muted, but please feel free to type questions in the questions for staff box throughout the presentation. And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Life's Lessons from a New eDNA Laboratory by Nicole Housley. And while Nicole shares screens, I'll share a bit more. Nicole Housley graduated from 2004 from the University of Mississippi with a BA in biochemistry and a minor in biology. Nicole worked 16 years at the University of South Alabama performing microbiology research before moving to the National Seafood Inspection Laboratory to become the contractor microbiologist performing analysis on animal byproducts for the Seafood Inspection Program. Thanks, Nicole, for presenting today. And I can see your slides a-okay, and I'll pass it off to you.
Thank you, Nicole, and thank you everyone for being here today. I'm looking forward to sharing our little DNA journey with you. And so let's start with who we are. We are the National Seafood Inspection Laboratory located in Pascagoula, Mississippi. We are the analytical laboratory who supports the USDC Seafood Inspection Program. Our microbiology team analyzes fish meal, fish oil, and other fisheries byproducts for bacterial and ruminant contaminants. And our chemistry team analyzes seafood for heavy metal contaminants and other toxins. So why eDNA? In 2020, our laboratory purchased an Illumina MiSeq to perform whole genome sequencing on Salmonella species that were isolated from fish meal for over a decade that we had stored. And we successfully completed that project and was looking for more opportunities to use our sequencer. Also in 2020, NOAA released an omics strategy that encouraged all laboratories to venture into new omics-based analyses with a focus shifting to eDNA analyses that will be essential to increasing sampling efficiency, ecosystem assessments, and having a decrease in environmental impacts. And thirdly, our laboratory sits at the Southeast Fisheries Science Center, which also hosts the NOAA CMAP Ichthyoplankton Survey Group, who so very graciously were willing to collect water samples for us for our pilot project in learning how to do eDNA. So essentially, you know, we have the resources. Oops. Sorry guys, it looks like my froze.
No worries, Nicole. Oh,
okay, there we go. All right. So we have our Illumina MiSeq, and we have the time and the people who were very willing to undertake this project. So our approach, you know, we gathered some papers, and what really caught our eye was this paper by Emily Jacobs Palmer on environmental DNA metabarcoding, that they were using DNA metabarcoding to look for species that contributed to harmful algal blooms, which is a definite concern. And in their paper, they targeted the mitochondrial cytochrome C oxidase 1 gene. The primers that were used were designed by Larray et al. In 2013 and are used for the detection of metazoa and have been successfully used on eDNA water samples, which is what we were targeting. And because we're also microbiologists, we were wanting to also look at the bacterial population that was in our water samples. And so we pulled the 16S metagenomic sequencing library prep protocol from Illumina. And in this, the primers target the prokaryotic 16S ribosomal RNA gene. The primers were designed by Clinworth et al. In 2013, and they target the variable regions 3 and 4 on this gene. And we've seen many papers where 16S has been successfully used on eDNA water samples. So we had our two targets, and then we moved into our sampling. And so in fall of 2023, the NOAA CMAP ichthyoplankton survey performed water collection for our project. At certain stations, they deployed this CTD with Niskin bottles. Samples were taken at the surface and at chlorophyll max and or the bottom, just depending on where the chlorophyll max was. 1 liter of water was then filtered onto two 25 millimeter 0.2 micron cellulose acetate filters using a standard vacuum manifold setup. Those filters were then stored dry in Whirlpack bags at -80 until the extraction process. For the DNA extraction, we were going to use the QIAGEN DNeasy blood and tissue kit with modifications that were similar to Walt et al. That we found on protocols.io. And this is a kit that our laboratory is familiar with, as it's what we use to extract DNA for our ruminant analyses. So in this method, we were going to thaw our filters in sterile Petri dishes, then cut those filters into thin strips. The strips would then go into tubes containing 2 different sizes of glass beads. We would then add a modified amount of the lysis buffer, beat those tubes at 45 seconds each for 2 rounds. Those tubes will then incubate at 56 degrees for 30 minutes after each bead beating cycle. We would then add a modified amount of proteinase K and then do an overnight incubation at 56 degrees with shaking. And then the next day would use a vacuum manifold to finish the extractions following the manufacturer's instructions with adjusted volumes to account for the adjusted volumes that we used for the lysis. So before we went to our plankton samples, which are a one and done kind of sample, we wanted to make sure we could work out our PCR protocols using this Kappa Hi-Fi polymerase that was recommended in that Illumina 16S protocol. So fortunately, a colleague of ours had a saltwater aquarium in her house and graciously brought us water that we could then filter onto the same types of filters that were used on the boat. And then we could extract the DNA from those filters and work out all of our PCR protocols before extracting the DNA from those plankton samples. So for the mitochondrial cytochrome C oxidase 1, we're going to call it the MCO1 and the 16S primers. We had read that a touchdown PCR was best for those MCO1 primer set. And so we performed a touchdown PCR using different concentrations from our aquarium DNA. And what we found is that we didn't get any product for our MCO1 primers, but got some product for our 16S primers at the lower DNA concentration. And so we definitely considered this a fail, not to get anything. And so on the next run, I called it my just in case run, because sometimes when you're doing something for the very first time and nothing works, you think, oh, what did I mess up? So I repeated the exact same protocol for the touchdown PCR with the exact same concentrations of our aquarium DNA and got the exact same results. So no product for the MCO1 primers at the 25 to 100 nanogram range. And we got product again with the 16S primers, but only at the 25 nanogram. So we started thinking that maybe we were putting too much DNA in the reactions and we were getting inhibition of the PCR, which is not an unheard of phenomenon with these types of samples. So again, a little bit of a fail. So what happens if we decrease the amount of DNA we put into our PCR reaction? So we followed the same touchdown PCR, and now we're just going to focus on these MCO1 primers and take out the 16S for now, since we were at least getting bands for those. And did a concentration gradient all the way down of our aquarium DNA, and again, nothing. So after delving into the literature some more, and delving into our kits, our PCR kits, that Kappa Hi-Fi polymerase, their instructions, we noticed that we weren't seeing a lot of papers using this polymerase. And like I said, this is a polymerase that was recommended out of the Illumina protocol. And so we said, okay, what does the kit instruction say? Maybe, we need to modify what we're doing based on this polymerase. And so the instructions suggested that we try a gradient PCR. And so basically we set up different concentrations with our aquarium DNA, and they were going to be the same PCR reactions. They were just going to have some that went through a 60-degree annealing temperature, a 66-degree annealing temperature, and some that went through a 72-degree annealing temperature, just to see if we got anything. So instead of using that touchdown PCR, we're just using this simple, straightforward gradient PCR. And guess what? We actually got some smears. And I've never been more excited to see a DNA product that didn't look like a DNA product, but at least there was more than there was. And so if you'll notice that we had to increase the concentration up to 100 nanograms to get even a decent amount of product. And so we wanted to run the protocol again, but tighten up the annealing temperature in that gradient. And so we're only going from 66 to 72 this time, to see if we could find maybe a sweet spot, and maybe we would get a little bit better clarity on our bands. So in this one, we had water, RO, so that was just laboratory water, so no DNA. We had algae, which is what we called our aquarium DNA. And then we had some samples right from outside our facility that the Coast Guard graciously supplied for us. And so we used some of that DNA because, we thought we could get more of that water if we needed to. And what we see is that, and we only used 100 nanograms. And so we see that our algae still gave us the smears, but we still weren't getting anything in our water samples that the Coast Guard collected for us. So, okay, maybe we need to increase the amount of DNA out of those Coast Guard samples. So we ran another PCR reaction. We chose 69 degrees for the annealing just based on the previous gradients that we had run. So we ran increasing concentrations of those Coast Guard samples, and we ran the 100 nanograms of our aquarium DNA as our positive control, because at least we were getting smears with that. Nothing. Now nothing's working. Not even the aquarium DNA is working. So why did our reactions stop working? So we phoned some friends. So we reached out to our friends Padmini and Amanda, who work at FDA CIFSAN, to discuss these problems that we were having. And so according to them, smears are not always bad. So sometimes they see that in their PCR. So it could be that we were on the right track, at least with that. But as to why the PCR reactions quit working, there could have been several things. We could have degradation in our PCR reagents, degradation of our DNA, or degradation of our primers. So now we needed to troubleshoot what was going on. And so the first thing we did was we isolated fresh DNA from our Coast Guard samples and ran that with our 16S primers, since we knew those would work. And then we also had a bacterial lysate from a Vibrio species from a project that we had worked on previously to use also, because that should work no matter what, as it's a bacterial lysate. And so we can see here that we used new polymerase reagents with our Coast Guard samples and got bands. And we used our old reagents with our bacterial lysate sample and got a band. So we're ruling out our PCR kits. So then we isolated fresh DNA from our aquarium samples. So we got more aquarium sample and ran the MCO1 primer set with the new Kappa Hi-Fi reagents in case that DNA was degrading. And on our gel, we also ran just some of our forward and our reverse MCO1 primer set to see if we could see anything. And I know it's probably not very clear, but we did have a little bit of staining with our forward primer, but absolutely no staining for our reverse primer. So we think we've ruled out the reagents. We isolated fresh DNA. and we're trying to look at our primer set. So we're still at a little bit of a conundrum of just, is it primers? What is it? So we reached out to Zachary Gold at NOAA PML, who is an expert in eDNA, and discussed with him all the things we had done, what was happening. So his suggestions were to go ahead and just get new primers because it did look like there was some degradation. He suggested that we try a lower fidelity polymerase, this Amplitac Gold. So the Kappa Hi-Fi polymerase that we were using is considered to be a high fidelity polymerase, so it makes less errors. And so, but the Amplitac Gold is still a really great polymerase. And then he really emphasized that for those MCO1 primers to run the touchdown PCR. So he said, okay, all right, we're going to do it. We got the new Amplitac polymerase. We got a new set of MCO1 primers. We set up our touchdown PCR, and we used our aquarium DNA and held our breath. And guess what? It still failed. But if you look down here, you can see that our nanograms of DNA, 100, 150, 200, so it was a whole lot, which is what we had to use with that Kappa Hi-Fi polymerase in order to see anything. And now we're not seeing anything at that concentration with our Amplitac Gold polymerase. So I said, okay, one more round before we call anybody again. We're just going to lower the DNA concentration of the aquarium DNA with that Amplitac Gold and see what happens. And so same thing, we ran that touchdown PCR, and now we have bands. So we lowered our DNA concentration down to 15, 30, and 60 nanograms. And if you can see past that 30 nanograms, we're not getting a product anymore. So we do think inhibition was happening at those higher DNA concentrations, and that's why we didn't see anything on the first go around. But now we have a band that's very clear. And so for those MCO1 primer sets, our band should have been around 315 base pairs. So here we have a product that is running, close to 300. So we were so excited and said, okay, now we have a plan moving forward. We're going to extract our DNA from our plankton samples. We're going to perform the first round of PCR with our MCO1 primer set and our 16S primer set. We would then need to perform a second round of PCR to add alumina overhang tags to the end of our amplicons. And then a third round of PCR to add our alumina indices set. And then we would be ready to pull and load onto our alumina MySeq. So by the time we got to the extraction portion of these plankton samples, our samples had been sitting in the freezer for 5 or 6 months. And remember, we stored them dry too. So we didn't know if that was going to have an effect. But so after extracting all the DNA from all of our plankton samples and from all of our Coast Guard samples, we ran our touchdown PCR for the MCO1 primers. We modified it a little bit to be more in line with what others are using for this Amplitac Gold. And we got product. We got product everywhere that we were supposed to get product. And that product is running close to 300 base pairs. So we were super excited and thought we were on the right track. So now we needed to clean those CO1 PCR reactions and add our alumina overhangs. And so now we went back to the Alumina 16S protocol for adding these alumina overhangs and used a PCR cycle and the Kappa Hi-Fi polymerase that they recommended. And you can see here, we got PCR product. These alumina tags add 50 base pairs on either end of the amplicon. So what was 315 should be now around 415. And as you can see here, these bands are migrating close to 400. So we thought, wow, we are, okay, we're on this. And we did this we mirrored this with our 16S primers, and they did the exact same thing. And so we were super excited. So now we have our amplicon with our overhang tags, and now it's time to add our indices, which would give us our unique identifier on our sequences after the fact. So We cleaned our tagged amplicons with Ampure XP beads. We did the last round of PCR to add these Illumina DNA UD indices set A. We cleaned those amplicons with the Ampure XP beads. We then quantified, normalized, and pooled 96 libraries together, and then loaded our library onto an Illumina V3 600 cycle reagent cartridge. Post-sequencing, we looked at some of the QC metrics that were generated by the MiSeq. So here's our run summary that happened, and what we're looking at is that last column, that Q30, and that is our Q30 score, which was in the high 80s, which according to our colleagues is really good for a metagenomic run. Usually Q30 is higher for whole genome sequencing, which is what we had previously done. And so we were happy to see that. The other metric that we looked at here in the blue box is our clustering density. Here we noticed that we may have some potential problems with our clustering density. So typically you would see clustering density between 1200 and 1400, and we're getting our clustering density around 1000. So this, so we could have had problems when we pooled, or we could have not had quite the right concentration. We were hoping to load 16 picomolar of our library, but maybe could have loaded a little bit less. But so for our first run though, we weren't too upset about this. So then for our sequencing results, our friends at the FDA helped with data analysis. So they used a program called bbmerge, which takes our paired in reads. So on the sequencer, we sequenced each amplicon that sat in the forward direction and the reverse direction, and the bbmerge takes those and pairs those, and then does some other metrics that I'm still, learning about, and to tell us how good our run was. So what I'm showing you here, this is a typical plot that the FDA sees when they do this type of metagenomic run that we were attempting. On the y-axis is the number of reads, and on the x-axis is the amplicon size of what was sitting on the flow cell. And you can see this nice distribution, this nice, bell shape, for lack of a better word, and which is normal. So even though you've amplified certain sizes, you're not always going to read the whole length of your amplicon, but you should still see that the majority of your read is in the region of the size that you're targeting. Unfortunately, this is what our plot looked like. So this is our Coast Guard samples with our MCO1 primers. You can see our number of reads. Number one, we have nothing resembling what the typical metagenomic run looks like. So we have very few reads, and we have very tiny amplicons. So our target for the MCO1, after you take off the overhangs and the indices, should be around 315. And here, this plot is only from 0 to 300, with a lot of our amplicon, with a lot of these reads being less than 300 base pair size. And so the same thing happened with our 16S, whether it was from the Coast Guard or the plankton samples. We had very few reads and very small base pair size. And for the 16S, you're looking for something around 400, and these guys weren't even getting to 300. And so based on our discussions with the FDA over what could have happened, their hypothesis is that the DNA that we extracted basically just shredded. It was just shredded into tiny fragments that we didn't even have fragments of the appropriate size. So if you want to amplify something that's 400 base pairs, you need DNA fragments that are at least 400 base pairs. And so they think that we were able to get amplicons made in the PCR cycles, that there was just enough pieces of DNA, long enough for the primers to sit down, and that the polymerase just filled in the gaps, basically. And so, what caused that? is unknown. Their hypothesis was the kit that we used. And so we are planning on trying and doing this again this fall. The plankton group will go back out, and they have agreed to collect samples for us again, but we're going to do a few things differently. We learned that we needed to add more QC metrics during earlier stages of this protocol. So post DNA extraction, post amplification, and verification of the library pool, things we should have done better. And so on this next round, once we do our DNA extractions, we plan to run our DNA on an Agilent bioanalyzer using the high sensitivity DNA chips to see what sort of size we get out of our extractions, where are our fragments pooling size-wise. And on the library pool. So before we put our pooled library onto the Illumina, we should have also run it on the Agilent bioanalyzer to verify that we actually had fragments of the correct size. After reading other literature and having more discussions, we're going to preserve our filters this time in a protective solution after they are collected. So we're going to use a solution called DNA RNA Shield. So on the boat, once they filter the water, they're going to put those filters in this solution before they freeze them. And hopefully this will help if there was any degradation, since we did store those filters dry and for 6 months. So maybe there was also some degradation on the filter before we could get to the actual extraction. And then we want to extract that DNA from the filters as soon as they come back, as soon as we possibly can. We're going to try a different DNA extraction kit this time around. So instead of using the DNA Easy Blend tissue, which has been very successful for a lot of labs, we're going to try the QIAGEN Power Water Kit. That was the next recommended kit. And then thanks to Zach Gold and some other eDNA libraries, we now have a better collection of protocols that are being used in other very well-established eDNA laboratories. And so basically we just learned, there was a lot we didn't know and a lot we didn't know to ask and a lot we didn't know to look at. And there's a whole host of literature out there, and, it can be a little bit confusing trying to decide on your polymerase or your protocols. So, but we're giving it another go thanks to our wonderful director and our colleagues who are more than willing to help. And so that's what we learned. So we're not giving up. We're going to go another round and hopefully we'll get better results this time. So I just want to thank, Kelly Dennett for being such a great greenlighting this project, Dr. John Bell and Patricia Rabideau for being just champions of the project for us, Amy Whitney and Abanda for giving me the space to learn new things, Glenn Zapfi for collection, Zachary Gold for all of his discussions and sharing of protocols, Padmini and Amanda for their discussions and doing data analytics for us, and then my teammates Shanara and Jonathan for just talking me off the ledge and helping me get through these projects. So thank you very much for letting me share this with y'all, and if you have any questions, I'd be more than happy to answer. Thank you.
Nicole, thank you so much for the wonderful presentation today. I think there are so many people that have been, and scientists and external to NOAA, that have gone through these very similar processes step by step to ensure quality control throughout each process of this very complex and complicated protocols, all the way from collection to extraction and sequencing and eventually bioinformatic quality control and analysis too. Thank you very much for sharing.
Thank you.
We will now open it up to participants for questions. You can type your questions and questions for the staff box. and we'll get started on a few of those. So none in there yet, but I will start us off. My first question, Nicole, for you is, could you detail a little bit more about the collection process of these samples? If I heard you right, you mentioned that they were stored at -80 after they were filtered. Can you confirm that?
Yes.
Do you know if that was aboard the vessel?
I think it was on board the vessel also, and then it was also in our laboratory too.
Right.
So they do have a small on the vessel that they went out on. They do have a small -80 that they were able, and you know, these little Whirlpack bags, I mean, they didn't take up any space. And so that was the advantage to using them, was just space concerns.
Mm-hmm.
But
And
now yep, sorry.
Go ahead.
Just now we're going to put them in little 5 mil conicals with the DNA RNA shield this time.
Right. And could you detail how much water was collected and filtered?
So we had 1 liter total over 2 filters, and we combined those filters together when we did the extraction, so that the DNA we were working with came from an entire liter of water.
An entire liter. Okay. And did you conduct replicates across?
We did not, because this is a pilot project. You know, we're just right now more focused on the learning aspect of just getting down our methods, you know, making sure to do, you know, that's our main focus right now. I mean, you know, obviously we want data out of it, just, you know, for the learning, but that's our bigger focus is the learning, because like I said, for us, you know, this was so new to our laboratory, and, you know, doing the whole genome sequencing was just so vastly different than this. And so that was our focus more than anything else right now, instead of, you know, so we set up replicates within the PCR itself, but we didn't have replicate samples during the filtering process.
Right, right. And something to note that maybe could contribute depending on the actual collections of the samples is possibly the DNA coming out of the leader itself, depending on the depth, turbidity, and the qualities of the water where it's being sampled as well.
Well, and we've also, you know, just talked to some people who've even seen a difference in what they've been able to detect based on the pore size of the filter, a 0.2 versus a 0.45. You know, like something they were looking at when they did 0.2s, they could never get that, but then they switched to 0.45s. You know, so I mean, that's the thing, is we're so new at this, and so we've taught, you know, we've spent a lot of time with the literature and tried to talk to a lot of different people to try to kind of nail down things we could potentially do better.
Absolutely, absolutely. Well, that's a great segue into our first question. We have 2 comments with this before we get there. The first is by Maria, who states, really great presentation. Thank you for sharing. We also had Glenn, who confirmed, yes, there is a -80 on the ship in which you were sampling. Alrighty, our first question is from Jeff, who says, thank you for the great talk. I learned a lot. Just wondering why the amount of reagents used for the QIAGEN extraction were adjusted during the DNA extraction. Was it because of the filter size?
Yes. So, you know, we read a lot of protocols, and I'm just still in my mind can't figure out how these people did it. So in the standard protocol for that DNeasy blood and tissue kit, it only has you doing a DNA lysis step in 200 microliters. So when you do that bead beading in those 2 mil tubes, you know, you want to make sure that the lysis buffer, you know, coats the entire filter, and so we had to increase that volume in order to get enough liquid to saturate those filters. So that's why so much was used.
Mm-hmm. Thanks. Thank you for the clarity there.
Yeah.
Another suggestion comes from Kelly, who asks, have you tried using mock communities and using those in concert with their environmental samples?
I mean, we didn't make a mock community. We were hoping that using the aquarium water would be, you know, because we know this aquarium, I mean, she's got fish and corals and all kinds of stuff in it. So we were hoping that, you know, that would replicate a little bit better as far as like we'd have water to filter, you know, so we could work on our extraction process and things like that. So, but it is definitely something for us to think about moving forward for these types of projects.
Undoubtedly. We have one final question before we conclude today by Patricia, who says, based on your experience now and in your opinion, what hurdles do you perceive exist in standardizing these analyses, in particularly under ISO 17025, which I can't say I'm familiar with.
So that is a, yeah, I'm familiar with that. I think standardizing is going to just be difficult. You would definitely need some sort of mock community standard that you could run alongside all of your analyses as a, you know, standardize. I know I put these species in, I can get these species out. We definitely need to think about having some sort of non-native DNA to do extractions on, to look at our cross-contamination potential, you know, to make sure that we are not having new DNA entered into these samples. So we've seen labs, you know, who will run kangaroo DNA through the extraction process and through the whole PCR process, things like that, because you know, kangaroo is not in the water. To look at that their lab practices are good. So, and then you have the bioinformatics pipeline, you know, everybody kind of has their favorites that they use. And so how do you make sure that when you do your analyses that you are doing all of the QC that comes with doing bioinformatics? You know, these pipelines, they all have different QCs that they're looking at. They have different things that are making them, well, this is good, this is bad, kind of stuff. But if one group is, you know, there isn't a standardized pipeline. I mean, I think that there are groups who are trying to work on that. And if that ever becomes a practice, I think that would be awesome. So, but yeah, so there's some hurdles, but you know, maybe they can be overcome, maybe not, we'll see. But definitely a lot more controls and QCs going on.
Most definitely, most definitely. And with that, Nicole, and I would like to announce to the audience as well that NOAA Omics is releasing their NOAA Omics technical portal as a growing resource for new and expert users alike. That technical portal, a link to it can be found on the NOAA Omics website in the chat. So go ahead and check that out for those that haven't seen it yet. It is in development, but also shared publicly currently on the website. So take a look in there. There's also on the NOAA Omics technical portal an area for feedback as well. So I encourage everybody to take a look there. With that, Nicole, thank you very much for your candid and presentation today of lessons from an eDNA laboratory. I think we all have a lot to learn from everything that your lab has experienced over the past year.
Thank you very much for having me.
Thank you. With that, thank you all for joining us today. Our next seminar will be on September 18th on autonomous samplers to scale environmental DNA observations across large and remote geographic areas of the ocean. It'll be by Christina Preston at Membari. It'll be a good one, don't miss it, and we'll be back next month. Thank you guys for participating today, and thank you, Nicole.
Thank you. Y'all have a good day.